Blunt end ligation, it can also be carried out by using another type of synthetic molecules that are called as adapters. So as I told you that कि adapters भी जो हैं वो commercially available molecules हैं और इनका एडवांटेज ये है कि इसका एक एंड ब्लंट होता है और दूसरा एंड जो है वो स्टिकी या कोहेसिव एंड होता है, तो इसके दोनों एंड ब्लंट नहीं होते. सो दिस synthetically synthesized molecule, that is adapter, it can also be used to ligate blunt-ended DNA like the linker. So this adapter molecule, as I told you that it contains one cohesive end, but at cohesive end the 5' phosphate it has been removed and it contains hydroxyl group. So the removal of phosphate from the cohesive end, it has advantage. एडवांटेज ये है कि जब फॉस्फेट कोहेसिव एंड पे नहीं होगा तो 2 या 3 या 4 या मिक्सचर में जितने भी एडेप्टर मॉलिक्यूल्स हैं वो आपस में लाइगेट नहीं कर पाएंगे. सो द रिमूवल ऑफ फॉस्फेट विल प्रिवेंट द लाइगेशन or the self ligation of the adapters molecule. So we will mix these adapters with foreign DNA. So one end in the adapter it is blunt and foreign DNA or the source DNA it is also having blunt ends. So blunt ends they will be ligated with T4 DNA ligase, and then the fragments that joined with the adapters, they are treated with certain enzymes like kinases so that we can introduce phosphate at 5' cohesive ends. Now it is ready to join with the vector molecule. For example, adapter molecules are available that have 5' overhangs or sticky ends that are digested with BAMH1. So vector can also be cut with BAMH1 so that the complementarity it is there between plasmid DNA and the source DNA. So how we can use adapters molecule for blunt end ligation? So we will discuss it with the help of a diagram. So this molecules it is an adapter. As I told you that at 5' end the phosphate has been removed and it contains hydroxyl group so that the adapter in a mixture they will not ligate themselves. So different adapters they will not join together, rather they will join with the source DNA. So the foreign DNA that contain phosphate at both 5' ends, it is mixed with adapter molecules that is having OH groups at the cohesive ends. So the DNA ligase in the presence of ATP, it will mediate the reaction so that the adapters they will be joined with the source DNA. Now it is treated with kinase. Reaction is ATP mediated so that phosphates can be introduced as at 5' end. So this is an adapter molecule. Suppose it contain this cohesive end that are similar that has been created with the digestion with BAMH1. So the vector molecule it can also be treated with BAMH1 so that the source DNA and the plasmid DNA it may have same sticky ends. So then they are mixed together and then they are ligated in vitro so that we can get a recombinant molecule. As I told you that it contains BAMH1 site, so this adapter it is the example of BAMH1 adapter. So it contains the restriction site for BAMH1 so that even after transformation we can isolate the recombinant molecule from the host organism. और इसको BAMH1 के साथ ट्रीट करने के बाद हम अपना फॉरेन DNA जो है या जीन ऑफ इंटरेस्ट जो है इसको रिमूव कर लेते हैं या एक्साइज कर लेते हैं. So this is one of the example of the use of adapter molecule with BAM-H1 cohesive ends. So, the linker molecules are adapter molecules. They can be used to ligate blunt ends. And blunt end ligation, so it is very well facilitated. The main difference between linker and the adapter molecule is that, as you may have noticed that, in linker, the both ends, they are blunt, whereas in case of adapter, the one end it is cohesive and the other end it is blunt. So, in both cases, after transformation, after replication of the artificial recombinant molecule in a suitable host organism, after treating with BAM-H1 or any other enzyme, we can excise the whole target or the foreign DNA. So, this is one of the advantage of the use of linker or the adapter molecules that can mediate the blunt end ligation.